Hello, I'm Fairlie Reese from the Mortazavi lab at UC Irvine. The title of my talk is Swan, a Python library for the analysis and visualization of long-read transcriptomes. Firstly, I'd like to highlight why long reads are so important in the RNA field. As we know, genes can have different transcript isoforms, such as the ones shown here. In traditional short-read sequencing, determining the full exon connectivity of the transcript relies on error-prone assembly methods. While we can recover that certain exons are seen together by looking at reads that sequence through exon-exon junctions, determining the full structure of the transcript is difficult to do. Long-read sequencing, however, allows us to sequence through the entire transcript, which is important as exon changes can lead to functional consequences such as protein coding differences or regulatory differences in UTRs. In our lab, we've developed a pipeline called TALON to process long-read RNA sequencing data from both prominent long-read platforms, PacBio and Oxford Nanopore. The output from this pipeline can be used as input to Swan. I'll walk you through how it works. First, common long-read sequencing artifacts such as microindels are corrected with transcript clean. TALON then assigns each read a transcript identity. Models that are found to be novel are given a novelty category, which I will describe in a moment. TALON then filters novel transcript models for those that are highly reproducible across replicates and for those that do not exhibit internal priming. Finally, TALON provides transcriptomes for each input dataset along with transcript-level quantification information. Here are the novelty categories that TALON can assign to any given transcript model. A transcript can be known, or a complete match to a pre-existing annotated model, an ISM, or an incomplete portion of a known model, an NIC, or new combination of annotated splice sites, or an NNC, where at least one splice site is never seen in the annotation. Once we've attained annotated transcript models, it can be difficult to determine what the difference is between them, as illustrated from this genome browser shot showing a complex locus of transcript models colored by their novelty categories from TALON. Being able to discern and understand the difference between models is not easy, and could potentially just consist of a difference in a few pixels on a screen. This shortcoming in transcript model visualization, along with the need for a tool to perform isoform-level analysis on long reads, motivated me to develop Swan. Swan uses a graphical representation of transcript models to store, compare, and visualize transcriptomes. It can also perform differential expression analysis on the gene and transcript level, detect isoform switches, find novel exon skipping and intron retention events, and provide unique and interpretable visualizations and reports, such as the one below. For this presentation, I really want to focus on how Swan represents transcript models, so I'll walk you through a short example. Here are two transcript models that belong to the same gene. These transcript models can come from a GTF or from a TALON database as output from the pipeline I mentioned a few slides ago. First, Swan assigns a node to each unique splice site. Here, you can see that there is a circular node for each splice site present in these two transcripts. These nodes are then added to the Swan representation, which will be shown below. Nodes are colored by their role in the transcript models. Blue nodes represent transcription start sites, orange nodes represent transcription end sites, and yellow nodes are internal to the transcript model. Then, we need to represent introns and exons, which we can do by adding edges between nodes. Green edges are added between splice sites to represent unique exons, which are seen in the above transcript models. When added to the Swan representation, they are arcs between nodes. We add introns similarly. For each unique intron that connects two splice sites, shown here in pink, we also add a pink arc to the Swan representation. This visualization model enables users to quickly notice alternative splicing events that make any two transcripts different from one another. For instance, exon skipping, as illustrated by the transcript models and Swan representation seen here, is easily recognized by an intronic pink arc that completely spans an exonic green arc. Similarly, intron retention is seen when an exonic green arc completely spans an intronic pink arc. Alternative splice donors and acceptors can also be seen when there are multiple incoming or outgoing exonic edges from the same node. I want to briefly highlight what we did with Swan for our pilot analysis. I ran Swan on 2 PacBio HepG2 replicates and 3 PacBio HFFC6 replicates that are lab deposited on the ENCODE portal. What we see here are 2 types of output graphs from Swan. Above is a gene summary plot for ADRM1 that showcases the complexity of splicing for the entire gene. Below, we see a specific transcript's path through the gene, where the darker edges and nodes are those traversed by this specific transcript. Splice sites and edges that are not seen in the annotation are indicated by outlined nodes and dashed edges. This transcript highlighted here contains a novel exon skipping edge as seen by the dashed intronic edge from node 4 to 11. Swan can also make gene-level reports such as this one, again for ADRM1. Each transcript's model can be plotted alongside quantification, novelty, and differential expression information. Shown here are each of the observed ADRM1 transcript models seen in the HepG2 and HFFC6 data, along with a heat map of their expression. I chose this gene in particular because not only does it exhibit novel exon skipping in the 4th model here, but it was also called by Swan as an isoform switching gene, which you can see in the differences in expression for the last 2 models between HepG2 and HFFC6. To summarize the rest of what Swan found in this data, it found around 4,000 differentially expressed genes and 5,000 differentially expressed transcripts. It also found 279 isoform switching genes, 1,021 novel exon skipping events, and 73 novel intron retention events. In the future, I want to apply Swan to other long-read datasets that are currently on the ENCODE portal, as well as to more recent single-cell long-read datasets. Additionally, I'm eager to improve the package with input from users as I only recently released it to the public. I'd also love to incorporate new analysis tools such as examining the complexity of transcript starts and ends, as well as ORF and NMD transcript identification. With that, I'd like to thank my lab, especially Dana and Gabby, who I've worked with very closely on long-read projects for the last 2 years. If you want to try out Swan, the URL is shown here in the bottom. Thank you for your time.